Hello, this is James Tai from Texas A&M University. Welcome you to the lecture series on single-cell high-throughput data science. I would like to thank Zhongxin University and Professor Zhao for invitation to give this lecture series. It's a great honor to be here. So these are the topics we are going to cover, and we start from a simple introduction about the history of single-cell technology and the main application areas, until more advanced topics such as construction of single-cell gene regulatory networks. So I will make the whole lecture series flexible, and I will invite my students to interact with me sometimes during the lecture, and also I will invite my graduate student to give part of the lecture, such as how I'm going to use Linux system to get the UML counts, for example. And yeah, so welcome to this 10 series of lectures. So my name is James Tai. I'm an associate professor in the Department of Veterinary Integrative Bioscience. I'm also affiliate member in the Department of Electric Computer Engineering in the Texas A&M University. And I'm running a computational system biology lab at the university. And this is my website. I will starting by introduce why the single-cell technology is important, why we're going to working on this around it. I often say that we are at the intersection of two major technology revolutions. One is machine learning, and another is single-cell technology. So this lecture series around these two topics at the interdisciplinary area of those two major technology. So when we say revolution, we mean the fundamental change of the field. So this is a single-cell biology a lecture series in called Spring Harbor about 10 years ago, and those are most of the biologists studying single cells at that time. It's very difficult problem because you have such little amount of RNA, mRNA in the individual cells. It's very difficult to study them. In the last 3 or 4 years, we have seen the breakthrough. So that's where the revolution really started. In 2018, the single-cell technology has been nominated as a breakthrough of the year of technology. And since 2016, when the introduction of the 10x genomics systems, then the paper published around the topic of single-cell technology applications have been increased dramatically. So we see this exponential increase of numbers of paper published using this single-cell technology. So 10x Genomics is a company in California, and they introduced this small device called the 10x Genomics Chromium controller. This is a device to help you to separate individual cells and prepare the library for them. So that's the technology used here. It's called a droplet. You see, the know-how of this technology is to use partition oil to capture individual cells to make them separate. Easy. Okay, so this technology is based on droplet using partition oil, and that's individual cells comes in. So you have from this droplet, And what you can see, each individual droplet, the structure is like this, with one cell attached. Okay, the cell is here. So you have some idea the size compared to the droplet, and also in the center, this is called the gel bin. Okay, on each gel bin, on the surface of the gel bin, there is covered by barcode, DNA barcode. Each gel bin has a unique barcode. For example, this particular gel bin will have A, C, A, C, T, T. Okay, so if this gel bin captured this cell and encapsulated by this droplet, so all the cell content, once it's licensed, will be labeled by this particular barcode, AC, ACTT. In a separate gel bin droplet, you have a separate, different barcode system. So this could be A, C, C, T, A. Okay, all the cell content, mRNA, will be labeled with this unique barcode associated on the surface of the gel bin. At the end, we will mix everything together, send it to the sequencing center to sequence. And at that time, you can use bioinformatics to separate individual cells by using this barcode labeled on the gel bin. So that's where the signal comes from. How can you separate them and also to still be able to do it high throughput using sequence? Because at the end you will mix all these thousands of cells after labeling by the gel bin barcode. As I said, there are huge impact on biological and medical research because this provides single cell level readouts for the signal from individual cells, which we couldn't do previously. So before text genomics technology become commercially available, most of people will use so-called bulk RNA sequencing to measure gene expression level in your sample. But now for the same sample, using this barcoding system, we will be able to read out all the genes expression. So if you think this x-axis genes, different genes, and the y-axis will be different cells. It wasn't like that. Traditionally, we get the information like this. For each sample, we have a vector. Of course, there are a lot of genes. But for each sample, you only have one data point for each gene. Okay, I will have a question now for Josh. Josh Cox. Have you heard about this RNA sequencing?
No.
Do you have some? Yeah. Do you have idea about this race mapping and to estimate the gene expression?
No, sir, I do not.
Okay. So I briefly mentioned, so you can extract mRNA from your sample, then reverse translate into cDNA and sequence them. By looking at, by count, how many rays is mapped to which gene, you can have a rough estimation of gene expression in your sample. It has been a very powerful approach to study gene expression in certain tissues and compare between disease sample or healthy sample. So now you have this high dimensional data compared to the traditional RNA-seq data you can get from each individual cell. So let's see how people using that to read out the whole landscape of transcriptome across many individual cells. Because this is a picture from the company. They're using single-cell technology to study disease mechanism. So this is a very good illustration because what you see is this, each dot here will be a cell. And this bar on the top indicate the expression level of some certain genes. If you know, you have annotation of, okay, we know this part are the cells contribute or associated with certain disease. You know that disease cells. You can start to look at the genes expression, give you a signature, tell you why this group of cells is so different from this group of cells. If you think this is a cancer cell population from one patient, some cells will be in the more say metastasis stage, and they are ready to migrate out of the location, in situ location to the other tissue or organs. But the signature will be important. And the single cell will tell you this information by only sample from one individual. Okay, we are not sequencing a lot of individuals. Traditionally, we have to sequence healthy individuals and, for example, cancer patients for certain tissue, certain type of cancers. But now you can use one patient's sample because some cells will be still in the healthy state, and some cells will be more disease related. You can have this translation.
Professor, on the previous image, that group that was to the right, like right next to the disease cells, is that just a group of known, like, pathogenic mutations in the cells, or what would that group be that's separated from the healthy and the transitionary cells?
Yes, for a certain question related to the cancer, people knows cancer cell will differentiate or divide it very quickly, and the genomic mutation may accumulate through this process. But more applications regarding to the state of the cell, you measure gene expression instead of a mutation in the genome. So that's a slightly different thing, but your idea is completely correct. If you follow a group of cell compared to another group of cell, and they separate for many generations, you will be able to detect mutation in their genome at the DNA level. But here, the picture may show mRNA level difference. Yeah, it's more like a transcriptome level instead of genome level. Very good question. So, yeah, so
excuse me, I also have a question.
Yeah.
Is this the single cell sequencing?
Yeah, you can do a single cell genome sequence or single cell mRNA sequence.
And this is from a droplet that they find all this?
Yeah.
Okay.
Yeah. So each point will be a cell, and you have all the gene expression information for this cell. But this particular plot here only show you one gene's expression. For example, this cell has this gene A expressed at this level, this cell at this level. You just cannot show some different genes. You have to just show one gene. But you can change. So I want to see another gene's expression. You have a different landscape.
Sorry, one clarification. So is every peak then like an individual gene within the single cell, and then the higher it is, the more it's expressed?
Yes.
Okay.
Yeah.
Got you. I'm following then. We're good.
Okay. So sooner or later, you will have time to play with real data. You will have a deeper understanding. How can you show this three-dimensional data points? You have to change. You have a position of a cell. You can put the position of a cell in 3 dimensions, and you can color each individual cell by certain genes expression level. If it's darker, then you think of gene A in the cell expression level is higher than a lighter color. And that's the data processing. I will show you examples later. All right, so let's just give you some concept. You can zoom in and see particular signals and compare in high resolution up to a single individual cell compared to its neighbors. You can still zoom in because each individual cell have a many different expression express genes. So you can look at different genes expression. This is just cartoon and not very accurate, but it gives you global impression of what's going on here. So another impact, direct impact on the field is people start to realize for each individual sample, there's so many different cell types. Okay. For instance, people study liver. And you know, the liver is very clean, defined organ. And if you cut a piece of tissue from the liver, we will think it's homogeneous. Every cell should be liver cell. However, once you do this single cell analysis, you will start to see different cell types. Okay. In this example, the hepatocytes, of course, majority of them will be hepatocytes. But you also see blood cell because tissue fill of this blood, and you have blood cell, epithelium cell, and you have all these different cell types you can read out from the loose homogeneous population of tissue. Okay, that's the first impression for most of the single cell study right now. Previously, you don't know the cell types is there, but now you found unknown cell types. You know, almost every tissue, every organ has macrophages. Macrophages behave differently. So you read out all those information. That's the power. Some disease may be only associated with specific cell types. So at this resolution, you will be studying cell type specific disease. Okay. This is another example to study the dynamics of the cell development from the very beginning of the fertilization. They use zebrafish. After 4 hours of fertilization, they sequenced individual genes in this embryo sample from zebrafish. After 6 hours, they sampled another embryos. They are not the same embryo, because once you dissociate the cells, this embryo has been destroyed. But what they can do is to sequence, process another embryo from the same breeding, and 8 hours, 10 hours, 14, 18, and 24 hours. Then they read out all the cell types and then generate this picture, superimposed onto each other, and also color them in different time points. So most of the cells are in the center when 4 hours after fertilization. And gradually, you see more differentiated cell types. You start to see neural crest. Once you have reached to 24 hours, you start to see mature muscle cells. It wasn't there when it's only 10 hours or 8 hours. By superimpose all this information together, you start to reconstruct a dynamic picture of cell differentiation through the fertilization and development of embryos. Okay, so far any considerations? So Josh, you have already Grant.
Oh, he's not here today.
Okay. Grace.
Yes.
Yeah, my question is, so far, what's your impression? It's a very useful tool, or just so-so? It's okay. People already know this. What do you think?
I mean, I think it gives a lot more information than like previous tools were available to give. So like you can get a lot more in depth on like the expression and stuff of like all of the cells instead of just, you know, like overall expression of genes. But like I honestly don't have very much background knowledge on this, but your presentation is giving me like a lot more information than I had before. So
okay, sounds good.
Does it provide a higher resolution of cellular differences?
Mm-hmm.
Okay.
Like theoretically track like the lineage of genes expression through cells, like all the way up to an adult zebrafish.
Yeah, exactly.
That's pretty cool.
Yeah. Okay, now I'm going to give you a demonstration of the real data, what it looks like. Okay, so this is data I played with for one of my collaborators today. Stop sharing this to give you a quick impression. So that's my desktop. I'm using MATLAB. So I have my collaborators send us the data we sequence in single cell gene expression analysis. So what we can do on the computer is to load his data. So I already made it clean and loaded this information. Can you see my MATLAB environment?
We can see it.
Okay, good. So you see, I already merged the data. So this is the data file. But I will teach you how to generate this. For example, you have this data, and now there are different variables. So big X. So I load this. I load this data in my computer. And you start to see this big matrix. This big matrix contains over 13,320 rows. That's a number of genes in 16,309 individual cells. So you can imagine this huge Excel spreadsheet is a huge number of cells and the gene expression level in there. So what are those genes? If you have this gene list, you see, okay, that's the name of genes. If I move down, you can see the gene name, AMZ1, AMZ2. So we have over 13,000 of them. All the genes, expression level. So the problem is, how can you view this huge matrix? I will mention later, you have a dimension reduction tool. So if 12 cells, see cell 1 and 2, as column indicate the cell, and the row indicate the gene expression of those genes in this cell. So the first position here is the first gene has no expression in cell 1. It has one expression in cell 6. Okay, then you can compare this profile between genes. See if between cells, if cell 1 and 2 are identical to each other, then your dimension reduction visualization will put them together closer in your picture. So I'm going to show you the picture for all the cells. Using a built-in function I created. So you start to see each point here is a cell. I color them into different colors because one is from a wild type of mouse, and another is from a gene had been knocked out in that mouse. So you have a knockout sample and a wild type sample. Okay, so I can rotate to see all the cells' relationship between them. If you see, okay, there's a small group of this. This small group of cells looks different from all other cells. They form a small clusters on their own. And then this application will go to search the database based on their gene expression, so-called marker genes expression, to tell you, okay, this is called a mast cell. So this is special cell types. And we can try another group, see what is the cell type there. So what I can do is select and drag this and say, okay, tell me using machine learning tool, okay, this is my macrophages. So I can do this quick annotation of what cell type are within my sample.
What would the smallest, like a typical cluster would be that is interesting?
Which one is the smallest? This one?
Yeah, would that be a cell cluster or?
Yeah, it's a cell cluster, right? I don't know because this is an unknown sample. Only my collaborators, they know where they come from, but I can try. I can say this is a neutrophils.
Okay.
Yeah, so there are high-level applications. You don't know what's behind those, the coding, but the function is there, and also this very nice interaction between. And what I can also do is to show the genes expression. For example, if I want to show expression level for certain genes, if I can select one of the genes and then pop out another. So this gene only show very expressed in very few cells. The gray means they are not expressed, and those yellow point means expressed. And this plot is similar like what I showed before. It's how high this bar indicates the expression level of that gene. So there is a small group of cells express this gene really high, whereas the rest of gene cells do not have the expression of this particular gene called ETL4. Okay, this gene only expresses in 1%, 1.5% of the cells. So I can try another expression of this gene. Okay, you see the relatively this gene expressed in most of the cells. 10% of cells have this gene expression. Okay, so for each gene, you have the picture like this. You have over 10,000 of these genes. So each one you can generate a picture like this. That's why I'm telling you, we have a high dimensional data. We just cannot visualize them all in one picture. So you have to generate a series of pictures. Okay, I have to go back to the slide to continue. So any quick comments from the audience? Yeah. Okay, so this is a slightly different view, but also give you impression of the variability of different genes among cells. So this axis is 1,000 cells, and this are the selected genes. So each row of this will be this gene's expression across this 1,000 cells, and the z-axis is expression level. Okay, the first impression you will have is some of the gene like this and that. Expression level overall is higher. This could be housekeeping gene. Each cell, every cell needs that gene expressed in order to survive. Some other genes will express more variably across individual cells. So what I say, this will be the gene expression variability. Have anyone think about this variability, gene expression variability for certain gene among cells? What the cause, what the potential factors to give this variability? Is that a clear question?
Can you elaborate more?
Yeah, so you see this difference. Some cell expresses gene high, some lower. Okay, so this is a variability. So what do you think? What kind of factors, or what's the reason you have this variability across individual cells?
Variations in cell types.
That's a good question. That's a good answer. Yes. If this 1,000 cells belongs to different cell types, for example, it's a mixture of B cell, the first 500, or some will be T cells. So B cells, T cells have different profiles for certain gene. You will create this. Yes, good answer. Any other? So that's a cell type heterogeneity. It will give you variability on certain genes expression.
There could be like changes of molecular traits, just like gene expression in different tissues.
Yeah, when you say different tissues, you mean the cell type difference. You still have the same, you still identify the same cause, the factors.
Yes.
It will be cell type or tissue difference. Yes, that's one.
Could cancerous cells have different variability?
Yes, that's a good one.
Could cells that are like in different stages of growth or division have different gene expressions?
Yeah. If cell is in the G1 phase, G2 phase, or M phase, S phase, they will have a different expression level for certain genes. Good answer. Good answer. So you think about this. And also, there are technique variabilities. Because just by chance, the randomness in the single cell sequence, their environment factors just cannot control this background noise. This noise will be especially strong in lower expressed gene because we have a capture efficiency problem because the starting point of the material of the mRNA molecule in individual cells is so small, the amount is so small. So there's high chance that you just cannot sequence it by chance. This is a more pronounced problem for lower expressed genes, for the genes like this. The overall level is so low. So the technical noise will have a huge impact on that. Because the total amount of these genes expression in individual cells is just so low. Okay.
Which you just have to have a lot larger sample base to be able to tell if, like, what level would just be normal background noise and what is actually being expressed.
Yeah, yeah. We will do this statistically because if you think that the noise is purely random, the signal will be different from the biological signal. You should be able to correct it. So as you mentioned, the statistics behind that, because you have to take into account if your background expression level for certain genes is too low, those genes, the measurement will be more pronounced, will be more substantially influenced by this technical. So around this single cell gene expression analysis or related analysis, there are many different packages that have been developed, different methods. I personally like to use MATLAB. I developed my own toolbox. Toolbox is a MATLAB function collection, and you have all the related functions in there. Most recently, if you work on the computation, scientific computation, people use Julia. It's another language, new language. So you will have a chance to play with all these clouds, command lines in MATLAB. It's very important to have this human data interaction. Okay, just like as I said, I showed you, there's no classification algorithm can give you the good capture of the different cell type you really need to be. I will have good examples to show you. You have to have this human data interaction. So this toolbox will help you to do some of this. Now, I'm going to explain the principle of a so-called pseudo-time analysis of single cell. So I will ask a question, see, barely from this plot, what you can infer, you can think about what I'm going to talk. Daniela.
Yes, I'm not sure what you mean, but it looks like you're just looking at different cells, DNA or RNA from like past generations, maybe? I'm not sure what this is.
Yeah. Okay. Good. So the pseudo-time analysis try to linearize the cells from a single cell sample, because when you sample a single cell, a group of cells, it's one time point. There's no further differentiation of. So if you want to infer what will happen for this group of population, you may want to look at individual cells variability, just like this picture I'm putting here. So if you want to study the gallop, the movement of the horse, but you don't have a video camera, what you have is a picture of a lot of horse. Okay, what you can do is to put the individual horse by their similarity and form a so-called pseudo-time series. Then you probably have some idea what's going on there. So similarly, for this group of cells, we cannot monitor each individual cells across the time because the sample has been destroyed. You only have a snapshot of the current state of the population of the cell. However, you can align the cells by their similarity to form a pseudo-time. Then you can use this pseudo-time as a covariance to study the change of particular gene or group of genes expression, follow the trend of this increase of pseudo-time. In that way, you learn what's going on, and you probably can use the result to predict if I have a cell at this stage, if you leave the cell, let it evolve or further differentiation, this cell may go this way and at the end look like this cell. Okay, so that's the principle behind the pseudo-time analysis. So this is one of the advanced single cell analysis method.
Professor?
Yes.
Do you ever see branching where you'd have two different branches or two different N cells?
Exactly. Very good question. We will create not only a single trajectory. Probably there's a branch effects. You will have multiple branches in your population. Exactly, yes.
I have a question.
Yeah.
Would you please explain pseudo-time one more time, if you don't mind?
Okay. So you put cells by their similarity, but in certain way you can align cells by their similarity, make them into this more linear form of this construction. So from this picture, you can assign, see, this could be the starting point of all the cell differentiation. Then you can follow this trajectory to construct a pseudo-time. It's not a real-time series data, but you create your own trajectory and assign each individual cells to the trajectory, different coordinates, to simulate or hypothesize they follow this trajectory to study the
the fate of the differentiation of the individual cell.
Thank you.
Okay. So let's give you some idea what we can do with a single cell data if you have one single cell data sample. Okay, we haven't talked about any comparison. This is just one person, okay? One sample. You have thousands of cells that's been measured. You can do dimension reduction, so you can visualize the similarity between cells. You can do this pseudo-time analysis. Okay, assign each individual cells a time point. See, if you think this will be time 0, then you move to time 1, time 2. You create this artificial time point, and here will be time n. Then you have your x-axis will be time from 0 to n. This will be time. And then you follow each individual gene's expression. Some gene will be lowly expressed at time 0, but highly expressed at time n. So you see, you detect those changes. Some gene will go opposite direction. It was highly expressed in cell at time 0, but it goes down at time n. Okay, we can move further to create so-called gene regulatory network to study the relationship between each individual gene among cell populations. So combine this network analysis with the pseudo-time. You can sample cells by their pseudo-time theory, and then construct a series of pseudo-time theory gene regulatory network. So once you have this data structure, you will be able to learn a lot of dynamics about the network. See, the link between these two genes was strong at time 0, but it become weaker, or other way around. There's topological change of this module of network through the time theory. Then you can learn all the parameters. So this is a high-level annotation analysis. Okay, so we can play with some of these functions using that tool. Yeah, so this is one small picture I took to let's see if I can play it. Yeah, so this is just a cell type annotation tool. I just show you the basic use of that already. And this video just gave you some impression. And you can just drag and okay, this alpha cell, this beta cell. So this sample is from the pancreatic islet. So you see alpha, beta, gamma cell, endothelium cells in this group. The most of them are the beta cells. So produce insulin. Okay, so this is another illustration that people use mouse as an example to study. We extract the neurons, cortex, and we extract the cell type. You want to study neurons, you get rid of all the epithelium cell, astrocytes, dendrocytes, oligodendrocytes. You extract neuron cells to create this trajectory. Okay, then sample cells. See, this cell group create this network, and then move on another sliding window, create this network. Okay, so this is just one example to use animal, extract cells, sequence them, dimension reduction to visualize cell relationship, extract cell types, do the trajectory analysis, do the network theory. Okay, higher level of machine learning algorithm can develop around this analysis. I gave an example here. So in this sample, we have 2 single-cell RNA-seq data. So this is abstract illustration, so you don't specify what this sample is, but any 2 samples, one from a patient, one from a healthy control, or wild type mouse, knockout mouse. Anyway, you have 2 samples. This pipeline will first subsample cells. Subsample means if you have 10,000 cells, each time you only study 1,000 cells, and then randomly sample another 1,000 cells. This is a way you can control for the huge variability or some single cell diversity. Okay, the noise, if you have 1 or 2 outlier cells, will have no impact on your whole pipeline performance. So you do this subsampling, but do multiple times. You have 10. If t equals 10, then you have 2 subsamples. Then for each subsamples, you create a regulatory network. Okay, the technology is called PC regression, principal components regression. This is a machine learning algorithm. Then treat this whole data structure as a tensor. Okay, tensor is a high dimension matrix. Then use the technology called decomposition to learn the major components from this tensor structure. So that's major components learned. Then you select top components and get rid of this least important ones to reconstruct the network. The purpose of this is a big noise. There's a lot of noise in there. But after that, we receive 2 networks, one from sample 1, one from sample 2. They are denoised regulatory network. Then we do something like called nonlinear manifold alignment to compare these 2 networks. And you mention maybe later what's the manifold. This is a data structure, data point structure. Okay, it's a concept in mathematics. Then once that, you can get the genes which change the regulatory relationship with other genes in the network. Then you can enrich those genes and see what's the function of those genes. In that sense, at the end, you will see, okay, in these 2 samples, what gene expression change really matters in terms of gene regulatory network change, influence a lot of other genes as well. So put them together, we have a biology hypothesis, then technology improvement, like single-cell RNA-seq technology, provide this unprecedented rich information, then plug your machine learning or artificial intelligence. Put them together, you should be able to test or validate your hypothesis, okay, to study the cellular transcriptome landscape, the map, the behavior of cells, okay, in the center, interpret and learn. If you really want to talk about the applications, we have a good application, see whether what's the best target of certain disease, which cell type we should target on, and which drug will have the impact on this cell type's gene regulatory network will change the fate of the cell behavior. Okay.
All right, this concludes our first lecture of the lecture series. I hope you had a better idea about what this single-cell technology is about and the potential applications and implications in the biological and the biomedical research. See you next time.